My parents are from this area, so I had breakfast with them in Mother's Day just a few days ago. And they asked me what I was doing in this area. They're not in the life sciences. And I said, well, I'm giving a talk at a pretty big conference. And my mom and dad said, oh, how big? And I said, well, I think there might be thousands of people in the audience. And they said, really? So mom and dad, this is for you. All right, here are my disclosures, the most relevant of which is that I'm a co-founder of Beam Therapeutics and Prime Medicine, companies developing base editing and prime editing therapeutics. The mutations in our genome that cause thousands of genetic diseases collectively afflict hundreds of millions of people and their families. So a longstanding goal of the life sciences has been to develop the ability to install, correct, or otherwise modify all possible types of pathogenic mutations so that we can study or potentially treat the broadest possible range of the corresponding diseases. Thanks to the visionary and pioneering work of many outstanding scientists, including the one you just heard from, Jennifer Doudna, some of whom are listed at the bottom of this slide, programmable nucleases, such as zinc finger nucleases, TALENs, and of course CRISPR-Cas9, really initiated the modern era of gene editing by enabling efficient targeted gene disruption or deletion. Programmable nucleases really excel at disrupting genes, exactly what Cas9 evolved in nature to do, because the primary response to cutting the DNA double helix in many types of cells is to initiate end-joining repair processes that rejoin the broken ends of the cut chromosome. Now, most of these end-joining events perfectly repair the cut DNA, regenerating the starting sequence. But a perfectly repaired DNA sequence can then be cut again by the same nuclease, so this process of cutting and end-joining occurs over and over until the end-joining makes a mistake that results in the deletion or the insertion of DNA nucleotides that prevent the altered sequence from being recognized by the nuclease. The result are indels, these mixtures of insertions and deletions that we can't control, unfortunately, but that usually disrupt the function of a coding or a regulatory sequence. Now, disrupting target genes can be very useful both for basic science research and for some therapeutic applications. For example, Cas9 nuclease has been used in a clinical trial to disrupt a gene that makes too much of a protein that causes transthyretin amyloidosis, resulting in a sharp decrease in the serum levels of the protein. And more than 200 patients to date, including Victoria and Patrick shown on this slide, have been treated in clinical trials with CRISPR nucleases targeting DNA sequences that, when disrupted, offer clinical benefit. Most genetic diseases, however, arise from mutations that have caused genes to lose their function and therefore can't easily be rescued by further gene disruption. Most disease-causing mutations therefore require precise gene correction rather than gene disruption in order to best benefit patients. And using programmable nucleases to precisely correct mutated genes in most therapeutic settings remains very challenging. Nucleases alone, of course, simply catalyze the cleavage of phosphodiester bonds and don't have the ability to make a specified change in a DNA sequence. In the presence of a donor DNA template, foundational work by Maria Jason and other scientists showed that double-strand breaks can also stimulate homology-directed repair, or HDR, which can correct pathogenic alleles. Unfortunately, the inefficiency of HDR in most cell types, as well as the downsides of double-stranded breaks and donor DNA template delivery, some of which you heard about in the last talk, have limited its therapeutic relevance. Double-strand breaks are now well established to induce undesired responses, such as chromosomal abnormalities, large deletions, and P53 activation. And as a result, clinical trials of gene-editing nucleases have thus far overwhelmingly been limited to those indications for which gene disruption or deletion can be therapeutic, rather than the much larger set of indications that could be addressed in principle by precise gene correction. So to enable efficient and precise gene correction in a wide variety of cell types, we developed base editing and prime editing. Base editors are shown here. They use the targeting mechanism of programmable DNA-binding proteins, such as disabled Cas9 or tail arrays or zinc finger arrays, to engage a desired DNA sequence. But instead of cutting the DNA, they use deaminase enzymes either taken from nature or evolved in our laboratory to directly convert one base to another by doing chemistry on the base itself. Then they guide the cell through the DNA repair processes that are needed to make this conversion permanent on both DNA strands. Cytosine base editors convert C to T or G to A, while adenine base editors convert A to G or T to C. Since nature does not provide any enzymes that are known to perform the chemistry needed by the adenine base editor, we actually evolved in our laboratory the first known deoxyadenosine deaminase enzymes in order to create the adenine base editors. We also developed, as you can see on the right of the slide, the first CRISPR-free all-protein base editors in collaboration with Joseph Mogus's lab that use tail or zinc finger proteins to target DNA, thereby enabling the first purposeful changes in the sequence of mitochondrial DNA in living cells and in animals. It had not been previously possible to edit mitochondrial DNA sequences because of the inability of guide RNAs, and therefore of CRISPR systems, to enter the mitochondria. Our lab and many others have since used base editors, both ex vivo and in vivo, to treat animal models of human genetic diseases. In collaboration with Mitch Weiss's team at St. Jude, we rescued sickle cell disease, the most common deadly monogenic disorder worldwide, in mice by using a bespoke adenine base editor to convert the pathogenic sickle globin variant in sickle cell patient CD34 positive cells to a naturally occurring benign hemoglobin variant called hemoglobin Makasar. And then we showed that transplanting these base edited cells into mouse models of sickle cell disease could rescue all the measured diseased phenotypes. And in collaboration with Francis Collins, Jonathan Brown, Mike Erdos, Leslie Gordon, and others, we developed an adenine base editor programmed to directly correct the mutation that causes progeria, the rapid aging disease, in vivo. We then used AAV9 in vivo delivery of this base editor to directly correct the mutation that causes progeria, the rapid aging disease. AAV delivery of this base editor profoundly rescued pathology, and as you can see from this Kaplan-Meier curve, greatly extends the average lifespan of mice, of humanized progeria mice. And here's what an untreated progeria mouse model looks like at 7.5 months old, which is about as old as these mice can live. And in contrast, here's what 3 progeria mice that each received a single injection of the base editor look like at 11 months old, much older than any of the untreated progeria mice can live. What about a genetic disease that, unlike sickle cell disease or progeria, is caused by a variety of different mutations, including deletion of an entire gene? Can we still develop a single gene editing treatment to serve all of those patients? Last month, we reported a therapeutic gene editing strategy to treat spinal muscular atrophy, or SMA, which has been the leading genetic cause of infant mortality worldwide. SMA is the consequence of 2 genetic accidents. First, all SMA patients have homozygous deletions or loss of function mutations in their SMN1 genes, which encode an essential SMN protein. Now, in animals, the loss of SMN1 genes homozygously results in the failure to complete embryogenesis, so there's no animal SMA. Humans, however, uniquely have an imperfectly duplicated copy of SMN1 called SMN2, which differs from SMN1 in a single C to T mutation that results in a missplicing event that makes a defective, short-lived SMN protein that's insufficient to support motor neuron survival. But it is sufficient to allow SMA babies to be born, only to suffer from progressive loss of motor function and typically death before the age of 2 in the most common form of SMA. 3 FDA-approved SMA drugs have really transformed the lives of thousands of SMA patients. However, these drugs do not fully restore SMN protein levels, don't allow endogenous regulation of SMN, which is thought to be biologically important, and require repeat lifetime administration, or in the case of the AAV gene therapy, is expected to wane over time but can't easily be redosed. So we set out to develop a one-time, hopefully permanent gene editing treatment for SMA. And rather than try to correct SMN1 mutations, which vary by SMA patient, instead we targeted SMN2, that imperfectly duplicated SMN1 copy that all SMA patients must have in order to be born. We tested more than 70 nuclease and base editing strategies targeting SMN2, changing the gene in ways that we thought might restore SMN protein levels. So on this slide are shown 35 nuclease strategies that generate indels that we hypothesized might stimulate SMN protein levels. And the most effective of these 35 nuclease editing strategies increased SMN protein levels by as much as 17-fold. However, SMN protein levels are about 40-fold lower in these SMA cells than in normal cells. So even the best nuclease editing strategies we identified did not fully restore SMN protein levels, in addition to carrying the other downsides of editing with double-stranded breaks. We also tested 42 base editing strategies. The 19 adenine base editing strategies shown on this slide all sought to convert SMN2 into a healthy copy of SMN1 by correcting that C to T mutation in SMN2. And the most effective of these base editing strategies fully restored SMN protein levels. We then packaged the most promising of these adenine base editors into a dual AAV9 vector and injected ICV, neonatal mice, SMA mice, which lack SMN1 genes and instead have human SMN2 genes. We observed efficient transduction of tissues in the CNS, including 43% average transduction of spinal motor neurons. And in the mouse CNS, the chosen base editor efficiently converted SMN2 to SMN1 in 87% of transduced cells. We assessed the rescue of motor function by electrophysiology. Electrophysiology assays revealed that ABE-treated SMA mice showed substantial rescue of motor neuron functional integrity, reaching averages similar to those of healthy heterozygous mice. And while adenine base editor treatment alone only modestly increased mouse lifespan, we thought about this and realized that the unusually short time window for rescuing these SMA mice, which only allow about 6 days for correction to take place before the fate of motor neurons is sealed, was likely too short for substantial levels of base editing to occur. In human patients, fortunately, the window for beneficial treatment is much longer, up to about a year. So we hypothesized that simple co-administration of a single dose of nusinersen, which is an SMA antisense oligonucleotide drug, together with the base editor AAV, would likely extend that therapeutic window for gene correction in SMA mice and would also more closely resemble the likely treatment state of SMA patients in a clinical trial. And indeed, the one-time co-administration in vivo of the base editor and nusinersen greatly extended animal lifespan from a median of 17 days untreated to a median of 77 days, along with a substantial rescue of motor function in behavioral assays such as the time the mouse needs to right itself after being laid on its back. Here's a video of a righting reflex assay for an untreated SMA mouse at 14 days of age, and you can see that these mice struggle to right themselves when they're laid inverted. In contrast, here are videos of adenine base editor treated SMA mice, again at 14 days of age, showing substantial rescue of righting ability. The beautiful work of scientists in other academic labs and in industry have advanced base editing into primates and then into clinical trials. Kiran Musunuru's lab and the scientists at Verve Therapeutics and Beam Therapeutics recently reported the one-time LNP-mediated delivery of adenine base editor mRNA into the livers of non-human primates, resulting in precise mutation of the PCSK9, of a splice site in PCSK9. Even though the goal of the study was to actually knock down PCSK9 levels, the scientists compared side-by-side Cas9 nuclease and base editors and chose base editors due to their observation of both higher efficacy and fewer undesired consequences. LNP-mediated ABE delivery durably reduced PCSK9 levels in monkeys by 90% and LDL cholesterol levels by 60%. At least 4 base editing clinical trials are now underway. Last spring, Wasim Qasim's team in the UK treated the first human patient in a base editing clinical trial using multiplex base-edited CAR T cells to treat T-cell leukemia. In late 2021, FDA granted IND clearance for the first US base editing trial, which uses an adenine base editor to install naturally occurring rare mutations in fetal hemoglobin genes that can treat sickle cell disease. And in late 2022, the first patients were dosed in an in vivo base editing clinical trial to precisely edit PCSK9 at that splice site to treat familial hypercholesterolemia. And at the end of last year, the very first clinical outcome from a base editing therapeutic was announced. This is Alyssa, a then 13-year-old girl in the UK who had T-cell leukemia and was given a very poor prognosis after both bone marrow transplant and chemotherapy failed to treat her cancer. She was given CAR T cells containing 3 base edits simultaneously in CD7, CD52, and the T-cell receptors. Following treatment with these triply base-edited CAR T cells, Alyssa's T-cell leukemia went into complete remission, and as of last month, 11 months after treatment, her cancer remains undetected. That it only took 6 years to go from the first base editing publication to the first positive clinical readout is really a testament to the work of more than 3,000 labs around the world who have received our base editing constructs from Adgene, resulting in more than 4,000 publications to date that include base editing. So base editors can make targeted transition mutations in cells, animals, and patients, but what about pathogenic mutations other than transition mutations? Since these other mutations account for about 70% of known pathogenic gene variants, we sought to develop other methods to directly install or correct transversions, deletions, and insertions, again, without requiring double-strand breaks or donor DNA templates. To enable precise gene editing beyond the mutations that can be installed by base editing, we developed prime editing. Prime editors are fusions of programmable nicases with specially engineered reverse transcriptases. They use an engineered prime editing guide RNA, or a pegRNA for short, shown in green here, which not only specifies the target site we wish to edit, but also encodes the desired edit. Prime editors nick the target DNA site, and then they use the 3' end of the freshly nicked DNA strand to prime reverse transcription of an extension on the pegRNA that serves as the reverse transcriptase template. The engineered reverse transcriptase domain of the prime editor then copies the desired edit directly into the target DNA strand, creating a 3' flap that the cell resolves into a heteroduplex containing 1 edited strand and 1 non-edited strand. The most efficient prime editing systems then nick the non-edited strand to cause the cell to remake that strand using the edited strand as a template, thereby completing the permanent editing of both DNA strands. Since the pegRNA's reverse transcriptase template is specified by you, the researcher, you can make virtually any local substitution, deletion, or insertion using this strategy. A hallmark of prime editing is indeed this remarkable versatility. Prime editing can perform all 12 possible types of DNA substitutions, as well as precise targeted deletions and precise targeted insertions for the first time in mammalian cells without requiring double-strand breaks or donor DNA templates. You can use prime editing, for example, to insert recombinase landing sites into the genome, shown in the lower right side of the slide, enabling large targeted gene insertion of multi-kilobase cargos. More than 95% of known pathogenic mutations are fewer than 50 base pairs in length, and thus are well within the size range for installation or correction with prime editing, which in its current form can mediate deletions of many hundred base pairs and insertions or substitutions of at least a couple hundred base pairs. Previous CRISPR-based editing methods derived their DNA specificity primarily from a single DNA guide RNA hybridization event. In contrast, prime editing requires 3 such events for prime editing to take place productively. And since each of these 3 hybridization events presents an opportunity for an off-target sequence to be rejected, we wondered if prime editing might inherently resist off-target editing more than a corresponding CRISPR nuclease. To test this possibility, we examined 16 known off-target sites in the human genome of 4 previously characterized guide RNAs, and a side-by-side comparison of editing efficiencies of Cas9 nuclease versus prime editors at these 16 off-target sites using the same pegRNAs to program both editing agents revealed that off-target prime editing was indeed much less efficient at all of these known off-target sites than with Cas9 nuclease. And many other labs in reports cited at the bottom of this slide have also investigated off-target prime editing, alterations of telomeres, changes to endogenous retrotransposons, altered splicing patterns, and altered gene expression using a variety of methods, including whole genome sequencing and whole transcriptome analysis. And these studies have also found prime editing to be highly specific for the target sequence, with no observations yet reported of Cas-independent prime editing consequences. Another important aspect of prime editing is that its edited products tend to be quite homogeneous, largely because the frequency of double-stranded breaks is substantially reduced compared to using nucleases. So in the examples shown here, prime editing can install this 3-base insertion in the human genome, and as you can see, the vast majority of the resulting edited DNA has the desired correction. This slide actually shows all the sequences observed at a frequency of 0.2% or greater. In contrast, you can treat with a Cas9 nuclease and an HDR template to install the same correction, and you get these results. So you get mostly a large mixture of these different indel byproducts that are typically all lumped together into 1% nuclease editing bar, but they actually consist of many different genotypes, each of which could, in principle, have a different biological consequence. To develop improved prime editing systems, we sought to understand the cellular determinants of prime editing outcomes. In collaboration with Professor Britt Adamson at Princeton and Dr. Jeff Hussman and Jonathan Weissman's lab, we used their RepairSeq CRISPR-I screening platform to knock down each of several hundred DNA repair genes in human cells and then quantitatively measured how each perturbation impacted prime editing outcomes. The strongest single family of hits from these screens were all components of the DNA mismatch repair system. Knocking down mismatch repair genes increased prime editing efficiencies and decreased the frequencies of undesired indel byproducts, consistent with the mechanism of prime editing that I showed earlier. But how can we evade cellular mismatch repair during prime editing? Well, in mammalian cells, mismatch repair operates primarily on heteroduplexes with just 1 or 2 mispaired bases. If there's more than 1 or 2 mispaired bases, the mismatch repair machinery will often fail to recognize that lesion as a mismatch. So we exploited this knowledge to evade mismatch repair during prime editing by carefully designing prime edits to purposefully install additional benign or silent mutations near the target edit, so that the prime editing intermediates natively evade mismatch repair. And indeed, we observed that additional silent or benign mutations near the target edit substantially improve prime editing efficiencies, and we now routinely use this strategy to design our pegRNAs. This advance came entirely from achieving a deeper basic science understanding of prime editing and how mammalian cells respond to it. We've also optimized codon usage, Cas9 sequences, linker sequences, and nuclear localization sequences to generate an optimized prime editor protein architecture that we call PE-MAX. And in work that I don't have time to present in detail, we also discovered that capping the critical 3' extension of pegRNAs with certain RNA pseudoknots yields ePegRNAs, as we call them, that resist cellular degradation, further enhancing prime editing efficiencies, especially in animals. When we combine these improvements, mismatch repair evasion, the PE-MAX architecture, and ePegRNAs, we observe a large cumulative benefit such that current prime editors with ePegRNAs generally outperform our original prime editing systems by more than an order of magnitude on average in both editing efficiency and in edit-to-indel ratios. These advancements in prime editing have had a profound impact on our ability to make therapeutic prime edits, especially in mismatch repair competent cells that used to be poorly edited. Here are 3 such examples, all of which started at low single-digit percentage prime editing, but now can be edited at potentially useful levels. The ability of prime editing to precisely replace one stretch of DNA with another raises the possibility of correcting the root cause of trinucleotide repeat disorders. In exciting recent data, the enhanced prime editing systems I described in this talk can precisely replace larger numbers of GAA repeats at the frataxin locus, which is the cause of Friedreich's ataxia, with non-pathogenic smaller numbers of repeats efficiently and with few indel byproducts. Scientists at Prime Medicine have applied a similar approach to cells from Friedreich's ataxia patients, resulting in the restoration of frataxin protein levels in patient-derived iPSCs and the rescue of axonal projections in dorsal root ganglia shown here, derived from the edited iPSCs. We've also used prime editing to replace a larger number of CAG repeats in HTT, the cause of Huntington's disease, with a non-pathogenic number in cells from animal models of Huntington's disease and also in mouse models. In a study we published just a couple of weeks ago that Kelsey Everett will present in her poster at this meeting on Friday, in collaboration with Jonathan Yen and Mitch Weiss's group, we used ex vivo prime editing of sickle cell disease hematopoietic stem and progenitor cells from patients to efficiently correct the sickle cell allele back to wild-type beta globin, now exploiting the ability of prime editing to convert a pathogenic T back into a wild-type A. And we rescued sickle cell disease phenotypes after transplantation into mice. 17 weeks after transplantation, prime-edited patient hematopoietic stem cells showed engraftment frequencies, hematopoietic differentiation, and lineage maturation that were indistinguishable from those of unedited hematopoietic stem cells, indicating that prime editing did not impair hematopoietic stem cell engraftment or differentiation in this model. An average of 42% of human red blood cells isolated after transplantation of the prime-edited cells from 4 sickle cell patient donors expressed wild-type beta globin, exceeding the threshold of about 20% thought to confer therapeutic benefit. Red blood cells from these transplanted mice after 17 weeks also carried less sickle globin and resisted hypoxia-induced sickling. These findings suggest the feasibility of a one-time prime editing treatment that offers a fairly unique combination of reverting the pathogenic allele back to wild-type beta globin without requiring any viral or non-viral DNA template. There's no DNA in this slide at all, and minimizing undesired consequences of double-stranded breaks. These results are also consistent with the data that Jen Gorey's team at Prime Medicine will present this week, reporting greater than 90% prime editing correction of chronic granulomatous disease patient hematopoietic stem cells, which were engrafted into mice, again, with no observed loss of engraftment potential. Researchers have recently described in vivo prime editing using a variety of viral and non-viral methods. For example, in work we reported last week, our lab achieved in vivo prime editing using iteratively engineered AAV, resulting in therapeutically relevant levels of prime editing in the mouse brain or liver, as well as significant prime editing in the bulk heart. And recent papers have achieved in vivo prime editing in animal models of human genetic disease. For example, Kai Yao's lab recently reported the use of AAV-delivered prime editors to rescue retinal degeneration in a mouse model of retinitis pigmentosa. And Gerald Schwank's lab used adenoviral delivery of prime editors to rescue phenylketonuria in mice. I'll end with a summary of the 3 major classes of programmable gene editing technologies that function robustly in mammalian cells. Nucleases can efficiently disrupt target sequences, but generate uncontrolled mixtures of indel products and induce undesired consequences of chromosomal cleavage. Base editors efficiently perform the 4 types of transition mutations without requiring double-strand breaks, but are vulnerable to bystander editing when a non-target A or C base is located adjacent to a target base. And older base editors can result in Cas-independent deamination of DNA or RNA, although the field has worked very hard to develop newer cytosine and adenine base editors that seem to avoid Cas-independent deamination. Prime editing can make virtually any type of local edit without double-strand breaks, with very high intrinsic DNA specificity, and with minimal possibility of bystander editing or Cas-independent editing due to its mechanism. But prime editors are newer, larger, and more complex than nucleases or base editors. All 3 of these technologies have now been validated in animal models of human genetic disease, and the first 2 have already entered patients in clinical trials, with prime editing anticipated to reach the clinic next year. Together, these gene editing technologies give me hope that one day we might have some say in our genetic futures so that we're not so beholden to the misspellings in our DNA. So to conclude, base editing enables precise target gene correction by allowing the 4 transition mutations to be installed at target sites without requiring double-strand breaks or donor DNA templates. Prime editing enables direct copying of edited sequences into target DNA sites, enabling a wide range of edits in human cells, again, without requiring double-strand breaks or donor DNA, and has been substantially improved through recent mechanistic studies and engineering efforts. And both base editing and prime editing have been repeatedly used, both ex vivo and in vivo, to rescue animal models of genetic disease, including sickle cell disease, progeria, SMA, genetic blindnesses, metabolic disorders, and others. As this molecular rendering of prime editing from David Belinsky's amazing team plays in the background, I'll just end by noting that not too long ago, the prospect of using laboratory-evolved molecular machines to correct a specific mutation in an animal to alleviate the consequences of a genetic disease really seemed like science fiction. So every morning, I wake up and pinch myself that I have the opportunity to work with the incredibly talented and incredibly hardworking group of graduate students, postdocs, and collaborators who made the work I presented possible. Former postdoc Andrew Anzalone, now at Prime Medicine, led the development of prime editing. Manda Arbab, who is starting her own lab at Boston Children's Hospital, and Janetta Matsuchek co-led the SMA work in collaboration with the labs of Arthur Burgess, Guanping Gao, and others. Peter Chen led the development of the mismatch repair evading improved prime editing systems in collaboration with Britt Adamson and Jonathan Weissman. Jesse Davis and John Levy led the development of AAV systems to deliver base editors and prime editors. Kelsey Everett led the sickle cell prime editing study in collaboration with Jonathan Yen and Mitch Weiss, and will be, again, presenting a poster on Friday at this meeting. Nicole Godelli, now at Beam Therapeutics, and Alexis Comore, now a professor at UCSD, led the development of our very first adenine and cytosine base editors, respectively. Luke Koblin, in collaboration with the labs of Francis Collins, Jonathan Brown, Mike Erdos, Leslie Gordon, and others, led the progeria study. Beverly Mock led the development of the CRISPR-free mitochondrial base editor in collaboration with Joseph Mogus's lab. James Nelson and Peyton Randolph led the development of the engineered PEG RNAs. And Greg Newby, now a new professor at Johns Hopkins, together with Jonathan Yen, Kaitley Woodard, Thiagaraj Meyeranathan, and the labs of Mitch Weiss and his colleagues, co-led the sickle cell base editing study. And finally, thank you very much for your attention, and I'm happy to answer questions.
I think we have time. We have time for maybe one question.
I think in a large audience, it's really hard to get a question.
Let's probably just move on. They already.
But you can all get really drunk instead, after you listen to the next talks. Oh, one question, all right.
Okay, one question.
You can just scream the question and I'll repeat it.
Thanks very much for the presentation. So my question is, you showed all of these structural developments in CRISPR. So what's next? So what's the next bottleneck that you can solve by engineering the protein even further?
So the question is, what's sort of the next big bottleneck that might be addressed in gene editing through protein engineering? And I think in terms of editing technologies, there's one obvious goal that many labs, including our own, are working on, which is how to directly insert large 10 kb genes into targeted sites in the genome so that we could prospectively replace loss of function mutations, regardless of their genotype, at endogenous loci, or install healthy gene copies at safe harbor loci. To do so with prime editing is possible and actually works reasonably efficiently. You can use prime editing to install a recombinase landing site and then use recombinase enzymes, which several labs have now shown, to install targeted genes. But to do so in a single step would also be quite interesting and potentially open up different applications. Although there's additional challenges involved in doing large targeted gene insertion, which include delivery of these very large pieces of DNA. So that's obviously one challenge. Another, which you've already heard about a little bit in Jennifer's talk and which is on everybody's to-do list, is to continue to innovate on the delivery side to try to maximize the target tissues for which we can deliver in vivo these editing agents. All right, thank you very much.
Thank you very much again.